So the main target of our session to work on tips for paper 6. So if you focused on the tips that we will talk about today, all of the tips needed, please let me know if there is any tip that I miss while answering any question. Okay, so we will answer an exam and we will check together if there is any tip missing for paper 6. The first tip, the tip for the magnification question is 3 marks question. And in this question, you need to remember the I am formula. And we said that I am formula is one of the formulas needed for paper 1, paper 2 for year 10, and paper 6 as well. So how could we calculate magnification? We will divide I by A. Image size divided by actual size. Type. What are the major tips for this question? You need to know how to convert from meter to micrometer. And we said several times in the class, like 1 millimeter equal 1,000 micrometers. So 1 millimeter equals 1,000 micrometers. So if you need to convert it to micrometer, it will be multiplied by 1,000. If you want to convert to millimeter, it will be divided just okay, just divided by 1,000. So you can convert between millimeter to micrometer, and it is extremely important in order to unify the units. In some questions, he might give you the actual size by micrometer, and you can't measure by micrometer, so you need to convert it to millimeter in order to calculate a magnification. Very important to know that the magnification will be beneath any figure provided. So sometimes بنقعد ندور على المagnification in between the lines in the word problem، لكن actually هو بيكون in a box كده beneath the figure, or um, already in 200x beneath the drawing or something like that. So it's easy to answer this question and there is no other tips. My tip for this question is to save time by measuring the Hamza in millimeter directly. Some of you will tell me, sir, I will measure easily in centimeter, then I will convert it to millimeter. It's totally up to you, but this is not an accurate method. افتكروا ان انا قلت لكم كده. This is not an accurate method to measure. So you should measure from the early beginning to save time and to get an accurate results by measuring in millimeter. اتعود يا ولاد starting from today while answering any past paper to measure in millimeter directly. ممكن وانا بميجر يا ألما in micrometer in centimeter اميجر ال line اللي عايزني اميجر or ال area اللي عايزني اميجر، مثلا let's say in centimeter هتكون 7 centimeter, but actually in millimeter it will be 72, not 7 centimeters. That's why I'm telling you, you should measure in millimeter in order to get an accurate results. Okay, this is the first step. Okay, uh, is the percentage increase and decrease. I will not say it will be a 3 marks question. It will be maximum 2 marks question. Okay, and you know this from paper 4 and paper 3 for year 9 and year 10. In order to get the percentage increase, it will be final minus original divided by original times 100. Percentage decrease, we will just switch the final and original. So original minus final divided by original times 100. So This is the formula for percentage increase and percentage decrease. You should show him you're working out and you should check what is the final reading and what is the initial reading or original reading. For example, if you have a data, Hamza, for 1900 and data for 1995, for example, and he wants the percentage increase. So you will check the data for 1995 and this will be the final, and the data for 1900 and this will be the original. خلاص, if you know well the formula that you will use, you can get the mark of this question. So this question is usually 2 marks question. خدوا بالكم احنا while taking a lot of tips هعرفكم ازاي تجيبوا ال 40 marks in this paper. For some students, this paper is somehow complicated paper and I cannot answer this paper. بس by time هتعرفوا يعني النهارده وبكره وafter tomorrow لو انت you practiced well على البيبر دي هتعرف ان هي it's just like a game. A practical paper to check your practical skills. The third, the tip for and very extremely important for a lot of questions. I can say this will give you up to 4 if you already know the variables. Variables in any experiment, 3. Independent variable, dependent variable, and control variable. What are the differences between independent, dependent, and control? What you are changing in any experiment, دايما كنت بقول لكم in the class, imagine that you are wearing now the lab coats going to carry out an experiment or investigation. So what you will change in the experiment is the independent variable. What you will measure after changing is the dependent variable. So what we will measure is the dependent variable. It should be يا كريم something measurable. ما ينفعش يا عمر تقول لي I will measure the rate of enzyme activity. أيوة يعني how could you measure this rate? You should tell me by measuring الgas production. Okay? You could measure يا عبد الرحمن gas production as a result of the enzyme activity, بس I cannot measure the enzyme activity في المطلق كده. مفيش حاجة اسمها كده. You cannot measure the effect of temperature on a specific reaction, بس you can measure gas produced in this reaction. وبased on this هتقول لي على فكرة يا سير, as temperature increase gas produced increase or not, or as temperature increase gas produced became less. يبقى لازم يا ياسين ابقى عارف كويس قوي ان what I am measuring is something measurable. حتى من اسمها it should be something measurable. ما ينفعش اقول له كلام in general كده في الانجليش وافتكر ان انا I answered the question properly. The independent variable should be one factor. With this is a very important tip. You cannot give him choices يا عز. variables. Independent variable is variable. For the dependent variable, what you are measuring, it should be also one variable. لو جيت كتبت لي في الإكزام 2 variables, it will be wrong. If you wrote in the exam 2 dependent variable, it will be also wrong. It should be only one variable changeable and one variable measurable in order to make a fair test. What about the control? The control could be more than one variable, definitely. For any experiment, you could find not less than 3 control variable easily. For example, volume of something, concentration or something, amount of water used, type of dye or stain used, diameter of something. So you could find a lot of control variable in this experiment, or in يعني I mean in this question for any experiment. My advice is not to write more than three. If he asked you to write the control variable, don't write more than three in order to limit the probability of having any mistakes in this question. عشان ما تجيش تفضل تكتب 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 one of them will be wrong. يبقى ليه؟ أنا I can save time. خدوا بالكم إن the duration of this paper is just 1 hour. There is no need to spend that much time in writing a lot, and all of these writing might reflect negatively, Maya, on your answer. This is tip number 3. Tip number is you have to memorize the 5 food tests that we have in the syllabus. Let's recap on the 5 food tests. The first one is the reducing using test. So reducing sugar test for glucose. امال انت ليه يا مستر قرفنا وبتقول لي reducing sugar? Because he might use this word in the question. ما تجيش يا سيف تقول ربنا يسامحك انت you did not mention the word reducing sugar. يبقى reducing sugar is the test for glucose. How could you carry out this test? By using Benedict reagent or Benedict solution. We should heat for 3 to 5 minutes. And if positive, I have to write this, ya Omar. If positive, results could be either orange, yellow, green, or brick red. All of the colors that I mentioned is extremely fine. But احنا already دايما بنكتبها brick red because there is no other tests give you this results. Okay, the next, guys, I cannot check the chat box regularly, so in between some tips كده, I will check the chat box if you want to write anything, and I will listen to all your questions. The second test directly is the non-reducing sugar test. Non-reducing sugar test is the test for starch. What is the test for starch? You should write using iodine, and it will be 2 marks because you will write the name of the reagent and the color. If positive, will be blue black. And we did all of these tests in school, if you remember. Okay, so the color will be blue black. Another tip for, or another test, the test for protein. The test for protein is the bioid test, and the test will give you a result as a purple color or a violet color or lilac color. All of these colors are accepted if you wrote one of them. The other test for fats is the emulsion test. What is the emulsion test? Emulsion. What is the emulsion test? In a test tube, remember with me, يا يحيى, this test tube. In the test tube, we will add drops of fats to the alcohol, because as you know well, any kind of lipids or fats doesn't dissolve in water at all. Even if you mix it for 30 minutes, it will split according to the density again. So we will dissolve lipids in alcohol, then we will mix them in 3 minutes in order to make sure that alcohol dissolves all of the fats. And the second step is to pour all of the amount in the test tube in a test tube containing water. What you will find is a white, cloudy, milky ring in the test tube. So we can say white ring or cloudy ring or milky ring in the test tube. This test called emulsion test. Don't be confused and write emulsification. because it is a common mistake, by the way, for year 10, because you took emulsification in the bile and the function of the bile, but here the test called emulsion test. The last test is the DCPIP, and it should be um, exactly like what I'm writing, capital letter, because this is an abbreviation for a big fat name of a reagent called DCPIP. This reagent for vitamin C test, um, you know well that we did not take anything related to vitamin C in the syllabus for year 9, but because this paper is a common paper between year 9 and year 10, you might find DCPIP or vitamin C test. What is the result of this test? It will decolorized or discharge or form colorless solution. السولوشن يا ياسين يا شرقاوي بيبقى الكالر بتاعه deep blue or purple. All of a sudden after adding السامبل اللي containing vitamin C السولوشن بيبقى colorless. Imagine بيبقى colorless. It's like a magic. And this is the test for vitamin C. الفود tests يا ولاد مهمة جدا. It might give you around 3 or 4 marks in the exam. لحد دلوقتي احنا almost we took around 12 marks out of 40 with questions. It should be totally direct for this. Tip number 5, then I will check the chat box if you have any. Is the tip for drawing. You know well the rubric for the drawing. You should draw the space provided. So السايز بتاع الدrawing يا حمزة is really important. Okay, ما ينفعش ارسم drawing in the same size as the drawing provided in your paper. It should be larger than the drawing that you have in the paper. Okay, لو عندي space like that for the drawing, you should draw using all of the space provided. Okay, وما تقوليش يا sir it will be really or extremely large. No problem at all. There is no shading. Any shading in the drawing will be wrong. There is no broken line. You cannot draw it as a broken line. This will be wrong. It should be unbroken line. For cells, don't draw any cells in your diagrams at all, except if he asked you to draw specific type of cell. Other than this, don't draw any cell. Yes, sir, I will take a full mark. Yes, you will take a full mark. Please, I'm begging you exactly the same as paper 4 and paper 3. Read the question well. بلاش نبقى خايفين من التايمينج وgo for the head of the question directly. اقرا الكويستشن وهقول لكم احنا اول ما ندخل الاكزام هنعمل ايه، بس خلينا نخلص التابس اللي needed. يبقى انا if I draw large drawing, if I draw unbroken line, if I draw any diagram without shading, if I did not draw cells بقيلي حاجة واحدة بس عشان اخد الفول مارك. is it possible يا سيلو ان انا اخد الفول مارك؟ طبعا طبعا، لان في ستودنتس بيقول لي سير فاكس no students will take الفول مارك في الدروينج. لا يا حبيبي this is a rumor. No, you can take الفول مارك if you draw a biological drawing. انت مش هتعمل لي drawing هاخده اعلقه في البيت. الدروينج should be a biological drawing، والbiological drawing has some certain rules. لو مشينا عليها you will take a full mark في الدروينج. And you can send me either privately or on the groups any drawing you want to check if this drawing like what Malak did today, if your drawing will be a full mark drawing or not. Please 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 don't draw any kind of cells. حتى لو الدروينج بتاعي يا ملك بيقول لي بس draw an outline for the layers of tissues. هتعمل الدروينج يعني in one of the exams الناس اللي حلت past papers وحلت معايا في الكلاسز هيكونوا فاكرين قوي الدايجرام ده. كان عندي دايجرام كبير قوي للليف والليف layers اوكي، وبعدين هو قال لي في الكويستشن draw an outline for this diagram، والستودنتس كلهم في الاكزامز وفي الاسسمنتس اللي كنا بنعملها throughout the year بدأوا يرسموا cells، a lot of cells, and they lost marks because he wants you to draw a layer for the upper epidermis, layer for the palisade and the mesophyll tissue, and a layer for the lower epidermis بس. قال لي حط لي الفاسكولار باندل، حطيت البوزيشن اوف ذا فاسكولار باندل. قال لي قول لي فين الزايلم وفين الفلويم في الفاسكولار باندل. as you know well في الليف الزايلم looking upward والفلويم looking downward. اوكي، فخلاص انا كده خدت الفول مارك للدروينج. سير احنا ما رسمناش حاجه، احنا we draw بس layer. Uh, we are just following the instructions in the question. Don't worry at all. Don't worry at all. بالعكس انت if you draw any kind of cells you will lose marks. وهنتشيك دلوقتي الدايجرام. يبقى انا in the diagram in the drawing I will take the full mark if I follow the rule. No shading, no broken lines, and the size should be larger than or represents like more than 75% of the space provided. The last point for the drawing, the label line. Your label lines should be that accurate, and you cannot draw a pointer or a head for your arrow. This is wrong. You should like a label line like this either بالruler or just a label line like what I draw now. It's perfectly fine. Okay, لو انا عندي cell wall وcell membrane, so you need touch the area that you want to label. أرجوكي لو cell membrane it should be like this. لازم أتاتش السيل membrane بالليبل لاين بتاعي. If I want to label for example a nucleus it should be like this. ما يجيش بقى يحيى صالح يروح عامل لي الليبل like this ويكتب لي nucleus. Wrong. It's unacceptable وهتلوز مارك بتاعت الليبل. Please read the question well يا حلا لأنه sometimes بيقول لي don't label وبخرج من الإكزام برقص بقى إنه هو أنا I will not label. في البارت بتاع الليبل ده some students بيقولوا لي sir shall we check all of the labels in the syllabus? Honestly for any drawing in biology you could label up to 3 labels من غير أي مذاكرة. يعني انت يا ألمى مش هتعرفي لو جالك السبيرم تليبل الهيد والميد بيس والتيل فور اكزامبل. لو انت يا عز جالك الليف ستراكشر مش هتعرف تليبل الابر ابيديرمز اللور ابيديرمز الفاسكولار باندل الباليسيد ميزوفيلير. لو انت يا عمر جات لك السيد والشوت والروت مش هنعرف نليبل السيد كود الشوت او البلوميول والروت او الراديكال. خدت الليبل 3 لاين. In the exam of 2017 يا حمزة كان في question بي ask you to draw السبايدر وليبل it. Some students رسموا السبايدر like this وعملوا لي legs like that. Is it acceptable? طبعا لا. طبعا لا. I have to draw it because it is an arthropod. I have to draw it like jointed legs. So we should do it like that in order legs. اللي رسم الleg like this خد المارك. اللي عملها just lines like this he lost المارك بتاعت الدروينج او part of the drawing marks. اوكي، ف whatever الدروينج was بس follow الروبريك اللي هو قايله لكم with الكويستشن اللي عندي. This is for the drawing tip. خلينا نتشيك الشات بوكس. What temperature يكندا do we heat at? For the Benedict there is no need to mention any temperature. احنا بس بنبويل. if you are asking for the Benedict. Does reducing Benedict also work for other sugars like maltose? He will ask you for glucose only. Okay. For this paper, he will not ask you for any other types of sugar. Glucose for a reducing sugar will starch polysaccharides in non-reducing sugar, but it will work. Okay. Yes, we
بس
will work paper 6 definitely. We will work on one of paper 6 now after talking about the tips. Yes. Who's asking?
أنا مش
انا عرفت انت مين يعني.
حاولتش إن أنا بس
ماشي يا محمد، اتفضل.
معلش يا مستر، واحنا ازاي بنرسم تيبل او لما بيطلب مني زي
حالا
جراف ولا حاجة؟
حالا احنا لسه ما قلناش التip بتاعت التيبل والجراف، لسه هنقولها حالا. انا قلت لك انا هقول لك 12 tips. ال 12 tips دول اتحداك ان يجي لك question في البيبر دي يعني، وما تعرفش تحله يا محمد. after this session بال 12 tips اللي هنقولهم انت لازم تقفل البيبر البراكتيكال دي ان شاء الله يعني. اوكي، خلينا نكمل احنا كده بس 5 tips. خلينا نكمل ال 12 tips. We are close to finish by the way. Tip the table tip 7 is for experiment you have, and he's asking you to draw put the independent side. هي دي يا جايز الحاجة الوحيدة اللي هتadjust معاك التيبل، انه if you start putting dependent what you are measuring on the left side التيبل عمره ما هيتظبط وهتلوز marks. On the left side of the table you should put independent. وبرضو يا حمزة if you did not write اليونت في التيبل I'm sorry you will lose 1 mark. Okay, and in the left side you will write what you are measuring. For example, if the independent variable for this experiment was time, so I will write time per either بقى minutes or seconds, it depends on the experiment. And if the dependent variable is the temperature change, it should be in Celsius degree. بس يا ملك يا حكيم أنا I have 2 sets of data for temperature change, A and B, for example. Okay, so no problem. I will split the heading into 2 subheadings and I will put all my data in. For the time, I will put 0 minutes, 2 minutes, 3 minutes, 4 minutes, 5 minutes, and at A, the temperature was 20, then 22, then 24, but here in B was 26, then it became 31, then 34, here it was 29 and 30 for example. التابل ده يديك 4 out of 4. Okay, because you put all of the independent variable in one column دايما في left side يا محمد، وفي right side you should write all of the measurable or the dependent variable. الداتا اللي طالعه عندك ايه؟ كل ما التابل هيبقى فيه extra task ده معناه ان التيبل هيكون up to 5 marks. وانا شايف honestly يا عمر ان يكون عندي table بكوبي just copy الداتا to put it in a table out of 5 marks ده انا خدت 5 درجات في جيبي if I did my table right. Okay, so this is extremely important how to draw a table. طيب وبليز read the question well. هو عايزك تحط التيبل بتاعت انهي داتا. Okay, put any data in a table in order to make sure that I added all of the data provided. Okay, محمد كده انت التيبل هتقفله لي ولا لسه في حاجة مش فاهم؟
إن شاء الله.
طيب ان شاء الله يا حبيب قلبي. اوكي. طيب التip كام؟ tip number 6. ال conclusion 1 mark. If 1 mark and I know well ان انا I can take المارك دي easily. There is a format for the conclusion. وخدوا بالكم عشان في student سألني قبل كده question. دايما ال conclusion بيجي ال question بتاعك بيقول لك ايه؟ Draw a conclusion for this experiment. في student قال لي ايوه مستر draw ايه؟ Okay. ال conclusion as you talk في ال English definitely several times before. ال conclusion دايما بيuse verb draw a certain conclusion for any experiment أو draw a conclusion of an incident or situation or something. فهو مش قصده هنا ان انت تدرو عشان ما تقعدش تقول لي يا مستر ده مديني space provided ادرو فين. اوكي لا هو قصده تكتب لي ايه الكونكلوجن اوف ذيس اكسبيرمنت. دايما الكونكلوجن بيتكتب بفورمات معينه يا سيلو. ايه الفورمات دي؟ دايما بقول له دي مثلا the higher the temperature the higher humidity. Okay, the longer the distance the lower the enzyme activity or less enzyme activity. Okay. دايما بنكتبها in that format. طب انا on which basis we can decide conclusion الداتا يا شناوي اللي provided قدامي. ما هو انا الداتا دي مش بلعب بيها. انا الداتا دي I have to study or to analyze الداتا. انا شايف من الداتا اللي provided for example يا شناوي انه ده كل ما التمبريتشر بتزيد الريت اوف الانزيم اكتيفيتي بتقل. لا هيجي عمر حاتم هيقول لي لا يا مستر انا مش شايف كده. انا شايف ان كل ما التمبريتشر بتزيد الريت اوف انزيم اكتيفيتي increase until we reach temperature معينة then decreases. مفيش مشكلة خالص. This will be 1 mark. Write the format as or the conclusion as you wish. بس أهم حاجة to use same format. اوكي وهنبراكتس دلوقتي على الكونكلوجن من الاكزام اللي we will answer together. اوكي tip number 8. tip number 8 هي الجراف. graph could be either bar chart graph or line of best fit graph. And this is a mathematical question, but honestly, I can take the 4 marks or the 5 marks of the graph. Number 1 يا عمر. هو عمر عبد الخالق؟ عمر عبد الخالق في الميتنج or not? علي you are in. Hi علي. وعمار meeting جيمس جدا.
مستر
مستر تكلمني؟
عمر يا حبيبي انا I'm talking to عمر عمر احمد عبد الخالق. هي مايا.
هو البار تشارت المفروض يبقوا انه قريبين من بعض ولا؟
هقول لك حالا يا مايا حالا هقول لك كل التبس اللي محتاجينها للبار تشارت حالا. ايوه يا مايا بحبك كنت مركزه كده معايا. برافو عليكي. اوكي طيب خلينا نقول ايه الروبريك و it should be your question من هنا ورايح زي ما مايا سألت كده. ايوه يا سير ممكن تقول لنا الروبريك بتاع البار تشارت or اللاين جراف. how could I get ال 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 full mark. اوكي حاضر. هقول لك حالا يا عمار احنا we will answer this paper October November 2022 variant 2. This paper that we will answer together. اوكي بس لما نخلص الريفيجن يا حبيبي. If you want this paper دي البيبر اللي احنا we will answer together. خلينا نتشيك together ايه الروبريك for the bar chart. Number one, like any graph, I have to put on the x-axis. حالا يا عمار هجاوب الكويستشن بتاعك. independent variable. And on the y-axis, dependent variable. ومعنى ان انا قلت variable على طول with no doubt you have to write unit. لان انت you know well يا مايا if I did not write unit I will lose marks. اوكي دي اول مارك هاخدها. Regarding البارز should take more than 50% of the grid. يعني if I draw all my drawing perfect بس I did not use more than 50% of the grid I will lose mark. لازم expand the intervals بتاعتي in order to take more than 50% of the grid. Even لو بار واحد بس يا مايا يexceed ال 50% ويعديها خلاص أنا كده في السيف سايد. This is the second mark. The third mark, the width of the bars should be the same. Okay? The fourth mark, the distance between the bars should be the same. حتى لو distance دي عمر, it will be like half slot or half square. I have no problem. The distance دي تكون قد ايه يا دانيا؟ بس أنا I have يا روتاج to mention إنه the distance دي should be the same. طيب امتى بعمل شيدنج للبار تشارت؟ اوعى تعمل شيدنج للبار تشارت لو انا عندي value واحده اللي I will represent. If I have two groups of data or two sets of data, I can use one shaded bars and one unshaded bars, and I have to draw a key for my bar chart. لازم اقول له على فكره examiner الشيدد دي كانت بتريبريزنت الميل فور اكزامبل والانشيدد كانت بتريبريزنت الفيميل او الشيدد كانت بتريبريزنت الانسكتس والانشيدد كانت بتريبريزنت البيردز. الشيدد بتريبريزنت سبيسيس A والانشيدد ريبريزنت سبيسيس B. اي حاجه ريليتد لكده. يبقى that's all what you need for البار chart. في ستودنت بقى هيقولوا لي ايه يا محمد احنا ما بنعرفش نعمل الانترفلز بتاعتنا كويس. The best way ركزوا بقى معايا عشان دي هدية من مستر ماجد. اوكي the best way to draw الانترفلز accurately is to look at the highest data, the highest value. Let's say انه ال highest value is 15. Okay, and you have 10 slots or 10 squares in the grid provided. Okay, or خلينا نقول 60 and you have 12 squares في الجريد اللي قدامي. Okay, هعمل ايه؟ I will divide the 60 by 12, so I know well now انه each square represented by 5. So 5, 10, 15, 20, 21. So now لما عملت الطريقة دي يا عمار أنا زي ما بنقول in Arabic إحنا ضربنا عصفورين بحجر واحد. Number one يا شناوي عملنا إيه؟ إحنا كده we are making sure إن إحنا we use more than 50% of the grid لأنه أعلى داتا هتكون فوق خالص. Number two أنا I will make sure إنه كل الداتا بتاعتي والبوينتس بتاعتي represented accurately on my graph. For the line graph I don't have any tip honestly غير إن هو لو line of best fit وأنا I have الداتا بتاعتي like this أنا I should draw a line. There is no need to pass by all of the data يا دانيا عشان البارت ده دايما بيقلقكم. Sir, should we connect all of the points if he asked me for the line of best fit? يا ولاد زيه زي الماث. اللاين of best fit بي pass through أو in between البوينتس. Mar asked me a question. ايه the difference between describing a graph and explaining? he will never ask you in paper 6 to explain something because this is a practical paper. But he might definitely ask you to describe the graph. Describe it means analyze. ومحدش يضحك عليك ويقول لك يا ابني ده reading للجراف. اوكي لا analyze. خليني اديك example يا عمار. graph like this. الجراف بتاع الانزيمز. اوكي and here ال pH وهي ال تعالى نقرا الجراف ده together. اول لو قال لي describe الريزلتس اللي في الجراف بتاعك وكان الكويستشن out of 2 marks، اول مارك هقولها الترند او الباترن اللي in front of me. So you will say as temperature increase the gas production increases then it decreases. ده اول حاجه انا شايفها في الجراف، ودي اللي كنا بنقول عليها في بيبر 3 وبيبر 4 يا اولاد الجنراليزيشن تكنيك. Okay, and the second mark you will tell me about the optimum and the reading of the optimum pH. هتقول لي ان optimum pH was for example 7 and the reading or the gas produced at this pH was for example 100 in describe. Okay, ما عنديش مشكلة يا كندا. There is no problem. لا يا كريم no problem. You should pass in between the points اللي closed دي. Don't worry. Don't worry. بس لو أنت عايز تكون في السيف سايد وتكونكتد ل one point at least connected no problem. بس لو هو اللاين مظبوط in between the points كلها don't worry it will be right. You can find a lot of questions like this. طيب لا no need يا كريم according to recent specification. بس انا بقول لك عشان انت تبقى مطمن ككريم يعني while answering no problem connected to at least one point مفيش مشكلة. اوكي هو كريم بس ask me في الشات بوكس يا ولاد should الجراف يباس through one point. I already drew it like that. بس if it's not possible مش هيبقى big deal. لكن هو بيقول لي في some marking scheme asked for this. انا ما عنديش مشكلة خالص لو انت قادر انك تكونكت at least one point fine. مش قادر والجراف مش مديني ال chance for this no problem at all. Okay, another بتقول لي what if حسب الداتا يا ليان حسب الداتا بتاعك ما تقلقيش. there is no need to start from الزيرو. الايديا ان انا اكونكت البوينتس كلها او at least يكون اللاين in between البوينتس كلها. Okay, no problem يا كندا. try to round it to the nearest whole number. Okay, so no problem at all. If you found انه مثلا the highest point is 27.3 خلاص fine we can tell ourselves انه the highest point for example is 28 and divided by the number of slots. Okay, that's all يا كندا if you cannot by logic to put ال value او ال intervals on your y-axis. كل اللي انا بقوله لك دي just tools to help you. بس ان شاء الله يجي لنا data direct واقدر put it on الجراف without any problem. Okay, this is the tip of the graph. Another tip بما اننا بنكالكليت او بنتوك يعني talking about ال data the anomalous results and انا بقول لكم I am expecting الكويستشن ده في الاكزام after tomorrow. ايه هي الانوماليس results؟ he will ask you what's meant by anomalous results. ممكن water please. ايه هي الانوماليس results؟ you should say الريزلتس اللي doesn't fit the trend or the pattern. والدefinition ده بيكون 1 mark. ask you for an experiment Just second يا ولاد, I will drink water. He might ask you for an experiment. How could we calculate the average? Okay, يا كيمو, يعني لو أنا عندي data for an experiment for food that was 27, and this one was 28, and this one was 13. Will ask you to tell him how could you calculate the average for this data. The question ده يا ولاد هيكون 2 marks question, because you should tell him that I will drop the anomalous results, then you will tell him how could you calculate an average. You will add the 3 other values divided by a number بتاعهم. So 27 plus 27 plus 28 divided by 3 اقدر calculate the average بتاعي directly. Okay, this is for the anomalous results. بقيلي بالظبط احنا كده وصلنا لتip number 9, 3 tips. Number 10 is the source of error and improvement. The question ده بيكون either 2 marks or 4 marks, مش بيجي in between. If he asked you to list 2 sources of errors, it will be 2 marks. If he asked you to list 1 source of error in specific step in the experiment and the improvement, it will be 2 marks. If he asked you for 2 sources of error and the improvement, it will be, for each one, it will be 4 marks. Okay, so I have to read the experiment well, because you will realize while reading where the source of error is. So the source, I will give you an example of the source of errors. I will give you an sources of error and improvement in the experiment. The first reading phase, for example, that in the experiment they used 1 measuring cylinder to measure different volumes for different solutions. So you should assign 1 measuring cylinder per solution to prevent contamination. If he used same pipette for different solutions, you should prevent doing this to prevent contamination. Okay, if a student trying to use or to cut dough, for example, into 2 identical parts, and she cut this by without calculation by her hands, mainly into 2 main parts using knife, definitely this will be wrong. We should use mass or balance in order to get the identical mass in between 2 dough in front of me. If he used small sample size, this could be a source of error definitely. هو احنا ينفع يا اولاد نعمل experiment على ولدين وبنت مثلا؟ It should be, in order to be a representative sample in the experiment, it should be large sample size, it should be random to be representative sample in the experiment. Okay, this is for the source of error. If he wants to control the temperature in one of the experiment and he used a water bath at the beginning of the experiment, مش هيحصل كده heat loss. So I have to use في improvement, I have to use thermostatic water bath instead. لأنه thermostatic water bath we are using to control the temperature. Another source of error, if he counted the number of gas bubbles released, is it accurate? If this will provide you with an accurate results, definitely not. He should use a gas syringe in order to give you an accurate volume. Okay, if he didn't use a measuring cylinder sometimes, so we should use a measuring cylinder. If he didn't repeat the experiment, this could be a source of error. We should repeat the experiment at least 3 times and take the average. Okay, so definitely now you know a lot of the source of errors. And please, if he asked you to go for a specific step in the experiment, ما ينفعش تيجي انت وتختار له error من step تانية. That's why I'm telling you, read the experiment well لانه will question well, because he's sometimes asking you for specific step to get a source of error فيها. This is the error and some ways of improvement in order to get 2 marks in this experiment, in this question, or up to 4 marks if he asked 2 sources of errors. And number 11. Investigation question. Question ده الكويستشن ده بيكون out of 6 marks. ودايما لما بشوف الكويستشن ده ببانيك. اوكي، ودايما بقول لنفسي no way ان انا اخد ال 6 marks of the question. However, it's easy to get the 6 marks in this question. If you followed my steps, you will get the 6 marks. Number 1, انه دايما لما ask me plan an investigation to check the effect of something on something، دايما اللي بعد ال of ده بيكون ال independent what we are changing، and ال after ال on ده بيكون ال dependent what we are measuring. Okay, so it's easy now to detect the variables. Another tip for the أنا لسه ما قلتلكوش ازاي هاخد ال 6 marks، بس another tip for this question. Okay, إني please check الexperiment اللي before الplan and investigation، لأنه most probably you will follow same pattern in your practical. Okay, please don't tell me إنه أنا I will talk about measurements. Okay, because this is just a plan يا ملك يا حكيم. هي حالة plan يعني إيه؟ يعني أنت اللي هتقول لي أنا بعمل إيه؟ أنا البلان بتاعتي إيه؟ بس أنت you did not enter in order to check this. Okay, so a plan for the experiment is the plan for the experiment. هنا نقول البلان بتاعتنا بقى بنكتب فيها ايه؟ نمبر 1 او اول مارك يعني يا ريتاش هي السيت اب. ودايما في السيت اب قول له ان انا I will use from 3 to 5 sets من اللي انا prepare. يعني for example, if he asked you to plan an investigation to check the effect of air temperature on بلاش اقول الانزيمز بقى on transpiration in plants. اوكي، loss of water in plants. هيا يحيى هتعمل ايه؟ لازم اقول له ان انا I will design my experiment by having 3 different temperature and plants from same species, same width of leaf, same type, as we said, same species or same type of plant and same cutting. اوكي يعني قاطعين نفس الديستنس. وهتقول له range of temperature, for example 10 degrees Celsius, and you will adjust the temperature by the water bath 15 for example and 30 or 25 and 40 for example عشان يبقى عندي acceptable range in between. This is number one. كل اللي قلته ده number one. No need to draw definitely a theme. أنا بس I'm trying to show you. A second mark for the independent and dependent variable. ويا يحيى يا صالح write it as what I'm saying. Independent and dependent. So you will the temperature change and the dependent variable is the volume of water vapor. لو أنا عايز أريح نفسي ممكن أقول له إيه؟ الماس change in leaves. لأن طبعا يا يحيى لما الليف هت lose water after a while مش الماس بتاعتها هتقل؟ Definitely. Okay, this is the dependent variable. Not third mark الcontrol. And we mentioned now 3 controls in this experiment. The fourth for the procedure and measurements. How could you carry out this experiment؟ هتقول له ايه بقى؟ انت هتعمل ايه exactly in the lab؟ Okay, I will wait for 1 hour for 15 minutes to check the effect of different temperature on the plant. And this is the procedure and the measurements. I will collect the data. I will measure the initial mass or final mass to get the difference. شغل مخك بقى وقول لي انت هتعمل ايه في البروسيجر والميجرمنت. Fifth mark, I will repeat and take the average. Or the 6 marks هي اللي في الكويستشن ده safety precaution. Because we cut the plant in this experiment يا يحيى, we should use a cutting board. This could be one of the safety precautions. You should also use goggles. You should also use gloves. Okay. وهي دي actually عشان نبقى خلصنا tips هي tip number 12 اللي هي safety precaution. لأنه sometimes بيجي لي question out of 1 mark لوحده كده. Okay. Any other خدناها, any other method خدناها كده, pretty sure ان انت you will get 40 if you want out of 40 لو انت you followed اللي احنا قلناه في التكست. خلينا نتشيك الشات بوكس. then لا يا ليان, she will not give you a mark. don't write ان انت you will record الداتا. يعني هي point valuable جدا, هيفهم الاكزامينر انك فاهمه how to carry out an experiment. لا يا براء, please don't write this point to record the data on a table because this is not a point give you a mark. It will, as I told you, it will show the examiner ان انت فاهمه how to plan, لكن مش هتديني extra mark. اوكي, so يبقى كده انا عرفت how to get المارك. Fine. خلينا نgo for الexam quickly. Do you need 5 minutes break ولا نgo directly? to save time? Could you please tell me in the chat box
Before
أو حتى
direct.
قولوا لي loudly؟
Break يا مستر break.
طيب اوكي fine break for 5 minutes وبليز مش عايز بعد ال 5 minutes الاقي الميتنج بقى 5 students only 5 minutes then انا I promise you ان انا اخلص البيبر دي معاكم في 20 minutes we will finish the meeting at 10 don't worry we will finish the meeting at 10 so 5 minutes وترجعوا لي تاني quickly خلينا نقول ان احنا at 9 خلينا نقول 7 minutes at 9:40 we will be in at 9:40 we will be in okay okay خلينا نتشيك this paper يا ولاد انا زي ما قلت لكم ان احنا we will take maximum 40 minutes بس to read البيبر والquestion to make sure that we can answer انا دلوقتي قدامي paper في الاكزام اول ما بدخل الاكزام بassign 5 minutes for reading the experiment very important this is a time saving process لو انت ما عملتش كده انت you will lose time in going back and forth throughout the paper في كل question انت هتيجي تحله خلينا نشوف دلوقتي ال 5 minutes اللي هناخدهم واحنا بنريد ال question واحنا بنwork on ال question ده time saving or not For question 1, some students investigated the effect of hydrogen peroxide concentration on activity of catalase. So I think we decided now an independent variable and the dependent one بس اتفقنا ان dependent should be measurable فخلينا نشوف how could he measure الاكتيفيتي بتاعت الكاتاليز اوكي يبقى الاندبندنت هو الكربون الهيدروجين peroxide concentration let's check catalase is an enzyme found in yeast cells the enzyme catalyzes the breakdown of hydrogen peroxide to form water and oxygen والreaction ده releases heat The students compared the activity of catalase in two different concentrations of hydrogen peroxide solution by measuring the change in temperature during the reaction. The student used a yeast suspension as a source of catalase. Okay? So, خلينا نتشيك الستبس اللي الستودنتس عملهم. Number one, aluminium foil was wrapped around the outside of test tube. The test tube was labeled A. Aluminium foil was wrapped about the outside of the second test tube. Test tube was labeled B. So he has tube A and tube B. وهو he wrapped the test tubes by aluminium. A syringe was used to put 10 cubic centimeter of hydrogen peroxide solution into test tube A. The temperature of the hydrogen peroxide solution in test tube A was measured and recorded as 20 degrees Celsius at 0 minutes. Okay? يبقى at 0 it was 20 degrees Celsius. The yeast suspension was stirred. 1 cubic centimeter of yeast suspension was then added to the hydrogen peroxide in solution hydrogen peroxide solution in test tube A and the stop clock was started. The temperature of the hydrogen peroxide solution was measured يا ياسين يبقى احنا ال dependent variable here هو ال measured temperature dependent variable what we measured in the experiment okay طيب a syringe was used to put 5 cubic centimeter of hydrogen peroxide solution and 5 cubic centimeter of distilled water in test tube B وعمل نفس الستبس يبقى I can say إنه ال amount of yeast suspension is a control because we use same amount يبقى أنا بتاعت الاكسبيرمنت ايه الاندبندنت، ايه الديبندنت، ايه الكنترول بتاعي في الاكسبيرمنت. اوكي، خلينا نتشيك اول question يا ولاد. اول question he's asking you to record الداتا اللي in front of you in a table. اوكي، فاحنا خلينا نتشيك التيبل بتاعنا اللي هنعمله هو 3 هو بير بريسيلز. دي كانت for two test tubes. test tube A tube B 2 3 4 with 20 and then write down a reading in a table. And the last reading was 29. So you will get the difference between the smallest reading and test tube A وتست tube B. يبقى الماكسيمم temperature change اللي هو كان 14 here and it was 9 here. ال 14 دي كانت what 34 minus 20. ال 9 دي كانت 29 minus 20. كده خدت كمان 2 marks، يبقى 2 marks و 3 marks 5 marks من غير أي تعب. State a conclusion for the results. Okay, look at the results بتاعتك. لما كان ال concentration higher the higher the higher أو ممكن أكتب له the higher activity اللي احنا we measured from الريدنج. عرفنا ان احنا we measured ايه ال temperature. Okay, then the test tube covered with foil in step 1. Predict and explain the effect of the results of not covering the test tube. If I will not cover a test tube, the rate of temperature loss will increase. لأن أنا هلوز the temperature easily from my experiment. Okay, يبقى I predict temperature will be less. Why? Because temperature will transfer because there is no insulator. مفيش layer insulated. خدت ال 2 marks. Another question. عايز 2 variables. احنا we kept them the same in this experiment. احنا we kept what the same in this experiment. خلينا نlook. احنا we kept the time intervals. احنا من 0 to 5 in both test tubes. احنا we kept the total volume of the solution اللي في التست تيوب ال 10 cubic centimeter. احنا we kept also الكنترول بتاعي اللي قلناه ال 1 cubic centimeter اليست suspension. So by reading the experiment يا حبايبي احنا قدرنا نجاوب كل الكويستشنز. شفت يا كريم احنا we are going that fast because we already checked every single step in the experiment. Okay, خلينا go for the next question. In different experiments, the students investigated the effect of pH, okay, on the breakdown of hydrogen peroxide. يبقى هو ما استخدمش ال concentration, he used ال pH. State the two measurements that the student would need to calculate الريت. أول ما أقول لك rate you should know إن أنت you will use التايم. وبعدين في بوينت تانية يا ولاد، بص على اليونت cubic centimeter. This will be the volume of gas produced. يبقى ال volume of gas produced والtime هما اللي هيكونوا ال measurements for this experiment to calculate the rate. يبقى يا يحيى يا صالح لما يقول لي calculate the rate of any حاجة لازم ابقى عارف ان انا هجيب الحاجة divided by the time. Again, if he asked you to calculate the rate of any حاجة يبقى الحاجة divided by the time عشان الريت هو time. Okay.
تمام.
Next question. Yes. Do you have any question يا ولاد؟
لا هو كنت بقول
ماشي.
لحضرتك تمام يعني.
طيب ماشي. ايوه عايز اسمع تمام بتاعتك يا حبيبي. اطمن كده اطمن. Am I a good teacher? Am I a good
أيوه
teacher يا يحيى؟
أيوه يا مستر.
بتفهم يعني مني؟
أيوه.
طيب طب ما تزعقش يا يحيى عشان ما اقوملكش.
حاضر.
اوكي يلا نجو for the next question. بصوا بقى هو في in this question I have in pH 8 anomalous results. Okay وهنتشيك ده دلوقتي. He did the same experiment but he changed the pH. Okay so خلينا نتشيك ال question. What's meant by anomalous results؟ الريزولتس اللي مش بتفت الباترن او الترند. ازاي الستودنت هيكالكليت الافريج؟ لازم اقول له ايه يا محمد يا مصطفى؟ انه هيدروب trial 2 ال value of trial 2 هيكالكليت by adding ال trial 1 و trial 3 divided by 2. هتشرح له ازاي بس he got ال average in this experiment. خلاص we answered ال triple I. عايزني احط الداتا بتاعتي يا ولاد على الجراف و it's easy to put الداتا بتاعتي on the graph ال pH and the average rate of oxygen production. اوكي عايزكم تجربوا تعملوا الجراف ده and then let me know if there is any problem. اوكي حد ask me a question يا ولاد على الشات؟ ماشي. Next question. Describe the effect of pH on the average rate even if you don't have the data in front of you. بس I can say إنه based on the data in a table. Okay أنا شايف إنه as pH increase ال rate of oxygen produced increases then after pH 8 it decreases again. يبقى هو الكيرف هيبقى زي الكيرف بتاع الانزيم activity direct. اوكي وهقول له ان الاوبتيمم كانت فين. اوكي then the student wanted to obtain a more accurate value of the pH at which the average rate of oxygen production was highest. Suggest further investigative work the student should do. Let student لما يبقى عندي problem rate او هو بيقول لك في الريت الهاي I should use more intervals. I should use very narrow intervals in between. يعني instead of measuring the rate of oxygen production عند 7 و 8 و 9 I will measure it at 7 750 8 50 and 9. Okay to get more accurate results وهي دي هتكون السكند مارك. اوكي كده يا ولاد انا جاوبت question out of 23 marks مش حاسس. اوكي let's go to the next question. Mr. in a line graph is the x-axis the dependent طبعا فأي جراف يا كندا الاكس اكسس هي ال independent والواي اكسس ال dependent. اوكي خلينا نتشيك quickly the second question. It should be اقصر. في ال question ده he wants you to draw a large diagram of the salary stock and show the positions of the vascular bundle on your drawing, but don't draw individual cells. And finally, label one vascular bundle on your drawing. ماشي يا سيدي. عايزني أليبل بس one vascular bundle in my drawing. أنا هصغر بس عشان أوريكم الدايجرام اللي I will draw. الدايجرام بتاعي. Okay. Then اللوكيشن vascular bundle. كون الدروينج in front of me يا ملك it will be more accurate. Okay. كل اللي انا عامله بس ده عشان البن مش عارف اظبطه، لازم اظبطه ويبقى closed vascular bundle. يعني انا يا مستر بالله عليك لو عملت الدروينج كده هاخد الفول مارك؟ يس، بس لازم اليبل one vascular bundle. الجرام ده خدت عليه كام يا يحيى؟ 5 marks. اوكي، وانت ما تعبتش في اي حاجه، ان انت بس يو فولود الرولز اللي احنا قلنا عليها. السكند كويستشن measurements عادي جدا، هحسب الماجنيفيكيشن، هميجر البي كيو in this diagram وبليز measure in millimeter. يا ولاد بتاعي موجود هنا الدايجرام الايمج divided by الاكتوال times لا not times، خلاص divided by الاكتوال اقدر اكالكليت الماجنيفيكيشن. لازم بس وهو ابن حلال مليمتر عادي. الاكسبيرمنت دي بس عندنا فيها ووتر، بس الووتر دي كان فيها داي فit became a solution containing dye. الداي دي اللي هي الصبغه، وبدأ يتشيك كمان عمل انفستيجيشن عشان اقدر اشوف ايه داي. تاني two methods. الديبندنت بتاعي الاير تمبريتشر، الهايت in a specific period of time، يعني الووتر traveled قد ايه in مثلا 30 minutes او 15 minutes for example، or هليب او همارك الهايت واشوف التايم اللي taken to reach this point. الطريقتين صح والطريقتين perfect. عندي الاندبندنت التمبريتشر، الديبندنت what we said احنا هنميجر ايه either التايم or الهايت. we'll control everything. we should control السبيسيس، الدايمتر، الامount of water, even the type of the dye could be a control variable. البروسيجرز ان انا بس wait for 15 minutes in order to check الابsorption او how the water travel through the celery stalk. Then I will measure the height or I will record the time. I repeat the experiment, I calculate the average. Last question here is to describe a method can be used to find out if samples of plant tissue contains reducing sugar or not. So in this experiment, we should write about Benedict as a food test. I will use Benedict. I will for 3 to 5 minutes using بقى water bath or something. Then if positive, the color will be brick red. That's it. Okay. خليني أتشيك آخر question. Can you say the answer again of what question يا براء? Of what question? Gation or what? do you think يا ولاد after this session do you feel إنه paper 6 will be yes حاضر will be way better لأن
Yes,
خلاص
sir.
عرفنا التبس وعرفنا how to answer this. In tomorrow's session and after tomorrow's session we will answer 2 exams Sunday و 2 exams Monday. If you have any question of any kind kindly ask me directly. السيشن بتاعت بكرة هتكون at 1:30 زي ما قلت لكم والسيشن of Monday most probably هتكون at the same time. So manage your time about the 2 sessions عشان تقدروا تذاكروا وانا معاكم على الجروبس if you want to send me any question. اخر part plan and investigation تاني يا براء هنقول انه السيت اب بتاعي ان انا I will prepare 3 sets in different temperature. دي هيكون وهيكون فيها of plan then different provided dependent which is either التايم or الهايت. يعني لو انا عندي plant like this يا براء انا بشوف يا اما اريا البلانت وبتشيك التايم taken عشان الووتر to reach this area or بعمل الاوبوزيت بحدد التايم اوكي اللي should be taken to او يعني بحدد التايم وبتشيك to what point الووتر travels وبميجر الهايت. شوفي انت both methods are acceptable. بشرح اللي انا شرحته ده في البروسيجرز then بريبيت الاكسبيرمنت وبكتب السيفتي precautions. Okay? Yes. Thank you so much for everything. See you tomorrow and after tomorrow at 1:30. Bye.
Bye, miss.
Bye يا
Bye,
حبايبي.
Mr.
باي يا حبايبي. باي باي
باي.
باي يا حبايبي.